It is my pleasure to introduce this week's speaker, Dr. Alex White from the Smithsonian Data Science Lab. Alex and I kind of go way back. So Alex was a graduate student at the University of Chicago in the College and Evolution program. And I served on his dissertation committee, which is when I was first exposed to kind of the cool, innovative stuff that it's up to. And so, yeah, so Alex graduated from E&E department at U Chicago in 2018, Trevor Price's lab. He went on to a postdoc at Smithsonian in the data science lab for 4 years, and that kind of transitioned down to this permanent position. And actually, there are additional field museum connections through there. So, like the data science lab was sort of during the postdoc was headed by Rebecca Dykow, who was formerly a postdoc, but a grad student at U Chicago, and then a postdoc here. So yeah, so a lot of connections with Alex and the museum, and of course Alex is very familiar with our bird collection. So his dissertation research focused on birds in different aspects. But I guess what I want to try to convey is that what really, I think, sets Alex apart in sort of the field of ecology, evolution, biodiversity, sort of science, is he has this great ability to see connections in methods, especially as it pertains to data science, from other fields, and see how they might apply to interesting questions that we as biodiversity scientists are interested in. So it's this kind of ability to kind of synthesize and see connections, interdisciplinary connections, that has really impressed me, Alex's work. And so without further ado, I'll let him tell us all about it.
Okay, thank you. As Rick mentioned, kind of a full circle moment. I remember sitting in this room as a graduate student, learning about new and interesting things in the world of the Field Museum. So very exciting to be invited just to participate here and tell you about the work that we've been doing. Like Rick mentioned, I'm at the Smithsonian. I'm part of a group that's actually embedded in the center of the Smithsonian, not embedded at the Natural History Museum. So we have challenges and problems that pertain to data science, artificial intelligence, strategy around data governance and management, and even our own advanced computing systems. And I've been lucky enough to be part of this group for a while that's helping develop those strategies for the Smithsonian. So you'll see some aspects of the work that I present. Mostly it's really geared toward biodiversity science. But the Smithsonian is really interested in how those methods can kind of extend across the different units of the Smithsonian. So within biodiversity, we're trying to make connections between the Conservation Biology Institute, the Tropical Research Institute, and the Natural History Museum. So hence the photo here of some camera trap work that we've been doing on the center. Perhaps I don't have to, belabor the point too much to this audience, but clearly biodiversity studies are increasingly relying on sophisticated technology to monitor and understand the patterns and kind of different aspects of biodiversity and how they're responding to a changing planet. So all of these different methods of data collection, like satellites, sensors that are on animals, or passive stationary sensors like camera traps or acoustic sensors. All of these are generating enormous amounts of data, and this data is both extremely useful for studies and can be processed to generate useful, interesting insights about biodiversity, but also it, is an enormous challenge for us to manage and understand how we're going to move from individual projects that might be using some of this technology to kind of whole systems or regions, and understanding how all of those data fit together to tell a story about the biology. So museum collections really are a kind of subcomponent of how all of these data are kind of moving through this huge data pipeline that is ongoing right now at tons of different NGOs, all of the different natural history museums which are generating data. We wrote this review it was published in 2022, and it almost seems outdated at this point because the amount of data that a natural history museum or a natural history institution might generate for the purposes of processing it with artificial intelligence technology or some sort of advanced computing technology has grown way beyond our set of 4 examples here, which was genetic data, some bioacoustic data, or samples, so specimens themselves from collection. But today I want to focus. I'm going to tell kind of a very focused story about one aspect of natural history collections, and that's how herbaria can be used to generate insights about kind of the unique biology of a system by leveraging this technology, which I will describe in depth, which we kind of generally refer to as artificial intelligence, deep learning, or even just very generally data science. And then I'll come back to this and tell you a little bit more at the end about some of the things that the Smithsonian is pivoting toward that aren't necessarily images that generate pixels, but other complex sources of data that are embedded in natural history museums that can generate unique insights. Okay, so taking a step toward the biology a little bit, I think this is probably a map that everyone is comfortable looking at and reflects some underlying knowledge they have about the biology of the world. But the number of species in any place on Earth really varies widely by latitude. And here's just a map of the number of terrestrial vertebrate species. And clearly there are many more species contained in the tropical latitudes than outside. Talking to a natural history museum audience, it's no surprise that this is a pattern that's seen across many different groups, birds, mammals, amphibians, particularly so in plants. So here, just kind of familiarizing you with some of this figure, because I'm going to show similar figures that have axes that look like this. The number of species is on the x-axis and the latitude on the y. And clearly you can see the number of flowering species in the New World much higher at the equator than outside. So explanations for that diversity gradient generally invoke what I would say 3 different and potentially non-exclusive explanations. The first is that there are differences in the diversification rates between tropical and temperate latitudes. So more species are just being created per unit time. We have more species. The second is that there is a difference in the age and area between the 2 regions of the world that we're comparing, the tropical latitudes and the temperate latitudes. So more species in a larger place because the tropics are actually older. They've been around longer, and so what we're seeing is a product of the history of the Earth. And the third explanation is that there are differences in the ability for these 2 locations on Earth to accommodate species, so that there are more species in the tropics because there's more opportunity, more ecological opportunity for those species to coexist. So that's what I'll focus on here for this part of the talk. I'm just going to dive a little bit deeper into how that might operate, and then we're going to look at how we use artificial intelligence and some of the methods that can extract data from specimens to evaluate this hypothesis. So here is just a theoretical way in which accommodation might operate. We have some resource distribution as the white curve, and then a frequency distribution of traits for 3 different species. If we wanted to add a species to this system, the trait values for the 3 species that we see would have to narrow so that adding the green species has kind of provided, or by narrowing the trait distributions for the 3 species that were there, this green species can now coexist. So that raises the question of whether or not tropical species are more densely packed into trait space. So the underlying kind of theory here relies on the idea that species that are similar in shape and size perform a similar ecological function. But species richness does not always kind of perfectly correlate to functional richness. And here's just a nice kind of fun example from a genus of tropical birds, the white eyes. Here are 6 specimens, museum specimens, each representative of different species in this genus. Clearly, these are very similar species. In fact, they're essentially allo species across different islands in the Indo-Pacific. But they're representative here of 6 species if we were looking at kind of a species richness curve for the region. But if we were to measure traits associated with how they interact with the environment, we might find that they were extremely similar to one another and representative in one function group. So the 2 questions, and I apologize, my PowerPoint got a little messed up on the PC, but the 2 questions that I'm going to come back to at the end of talking about this application is, does morphological shape diversity vary according to latitude, and how does it do that? And then how do species at different latitudes then subdivide ecological opportunity differently? And like I mentioned, we're using artificial intelligence methods, and specifically a subdiscipline of that, which is computer vision, to quantify and summarize aspects of biology of organisms in a museum collection. So first, I'm going to talk kind of at length for a little bit about these methods, because I think they're really applicable. The approach itself is really something that I think is a paradigm that can be applied across a lot of different systems. And then we'll get back to some of our results and the insights that we drew from them. Okay, so we focused our study on ferns and the complex diversity of the shapes of their leaves. Here are about 13,000 different species distributed globally for ferns, give or take some. And here is 9 representative examples of some of the images that we have in our data set for ferns. So our data set does as best as we can to kind of capture the diversity of shapes of ferns, and I'll get into some of the numbers in a sec. So fern species numbers really do show the characteristic latitudinal diversity gradient. No surprise there, but here's just a map of kind of cleaned up data from GBIF, looking at all of the different specimens, about 1.4 million museum records on GBIF. The Natural History Museum at the Smithsonian is about 250,000 or so. I'm sure there are field museum records in there. And here's just another map showing that number of species and where they're distributed latitude. Ferns are very well represented in digitized collections, and for those working in the botany department or have seen, those of you who have walked through their halls recently, this might look familiar. I think they're digitizing in a similar manner the botany collection here at the Field Museum, but this is how all of those different collections or different specimens were digitized at the Smithsonian. That was a project that was completed 3 or so years ago and was a digitization of about 3.8 million records, let's say, individual herbarium sheets. So we took for our approach all of the available iDig Bio specimens for ferns and lycophytes. This allowed us to get, of course, our hands on more than just the images that we had in our digitized data set. There are about 513,000 images that we could actually resolve toward a reasonable taxonomic identity. We have about 8,000 species, or 8,600. So that's 67% of the global diversity of ferns and 320 genera. So a lot of the generic diversity for ferns represented in this data set. Of course, you can see just how different the shapes of these organisms are. So like I said, we're taking a deep learning or computer vision based approach to understanding fern shape. And the way that we did that was by building something called a convolutional neural network and trained it to label fern specimens with taxonomic identity. So without getting too deep in the weeds on what a convolutional neural network does, it essentially takes an image as input and it processes that image through a series of matrix, multiplications and functions, and produces at the end of a vector of probabilities. What is the probability that the input is part of any number of classes in the end? So we end up with a probability vector for the number of classes in our data set. We used about 2,000 species to train this data set because we needed a training data set that included multiple representative specimens per class. So we had 8,000 species at the beginning, but we needed enough training data for our model to operate. So we have about 2,000 species. And with that, we can evaluate the quality of our identification network by looking at how well it is capable of predicting a species or a genus, some taxon, when we already know the answer. And so we represent the output of that type of analysis in a figure like this. This is called a confusion matrix. Essentially, for a perfect model, every time the prediction is made, it should sit right on the diagonal where the actual genus is present, so that we would have 100% of the predictions fitting into the actual genus bin. So the best thing would be to see a blue diagonal and off diagonal elements all being yellow. The cool thing here is that actually the confusion, kind of hard to see with the lights here, but the off diagonal elements tend to fall within families. So when this model is confused about which species is the right, or which genus in this case is the right identity, it tends to choose another genus within the same family. So that's just showing kind of how confused it was about these 3 different species of tree ferns. So we were encouraged. It was a very accurate model. At the generic level, we were able to get about 97% accuracy. But there's a lot of data on these slides or on these images that isn't necessarily all that useful for us from a biological perspective. It might be influencing the way that artificial neural networks or convolutional neural networks make a decision about which species is which. And so some examples of the data that we are throwing into this model at this point are the taxonomic labels, the various institutional markers. And so is it picking up on something actually associated with that, making good predictions based on it? So we developed a quick kind of method for masking all of the plant pixels in any one of our images by developing another neural network which actually identified whether or not a pixel was plant pixel or anything else on this image. And that was a collaboration with BYU and some undergrads who developed this data set of high resolution differences between which pixels were which. And so they had to clean this up. And once we got a really good training data set, we were able to train a network to actually produce images from images that come in on the left to something that looks like this on the right. And we were able to then do that for 500,000 images and process those and go back to our model. And instead of passing in images with all of this other data on here, we were able to just work with the plants. So because we were just working with the plants, now we're kind of interested in more about what those images represent in terms of the shapes of the plants. So we continued to train a model using this data and actually ended up with a species level data model so in this case, it's representing 2,000 or so species. The accuracy of our model in choosing the exact right species was about 81%. It doesn't sound so good, but we're talking about 2,000 species of ferns. And a lot of the differences between these species aren't necessarily even evident on an herbarium sheet from the scale of what we're taking a picture of here. So we also looked at the species being present in the top 5 choices that our model made. So because it produces a probability vector instead of just a single probability for the maximum likelihood for a species, we can look and ask, is the right species in the top 5? And in that case, we're at 95.6% probability. So pretty accurate to the level of figuring out on some level, we understand which species which in these pictures. So these models, they take in an image and they produce this classification layer. Well, the interesting thing is, of course, because the image just contains this information, it can be reduced to a single layer in the model that actually represents, as best as this method can produce by this training process, a vector of the shape information, the features that are relevant for making the classification decision. And that is a body of data that is contained in that shape layer that we then took to kind of explore based on our questions about shape and how it varies across the globe. So I'll quickly breeze through some of this because this kind of can be somewhat cumbersome, but essentially we have an arbitrarily large shape layer. That layer, for no good reason, and basically because of the way that the math is built, what we would call the architecture of this model, that layer is 2048 in length. But it really is it's just an arbitrary size. And so others in the world of artificial intelligence and deep learning science have been studying whether or not those the size of any layer in this model has some intrinsic properties rather than the arbitrary properties that a mathematician chose. And in doing so, they were able to find that while these layers tend to have really high dimensional size, they actually have these intrinsic dimensional sizes that are very low, meaning this is kind of like the idea of principal components analysis. We have lots of data that goes in, but maybe few principal components that represent the total diversity in the data set. And we can use those intrinsic properties or the intrinsic components to look at diversity. So we did just that. I was able to model, the total amount of data contained in our data set, which was a, 500,000 by 2,000 data matrix, and estimated that it has the intrinsic dimensionality of like 8 different underlying features that really describe the total diversity in the data set. And using methods that are similar to principal components analysis, we then projected all of that data into low dimensions to actually look at the distribution of how those different specimens are related to one another. So here we're essentially looking at, not where it says landscape right here. So we're looking at a morphospace from our model that was used to generate species IDs. And in this case, each dot in this, they're hard to see here, but there are dots on the scatter plot. And the colors here represent the density of different specimens in the data set. So each dot is then connected to an actual image. So we could say that an image containing or a species or a specimen that's processed and has these coordinates is represented by this image. And clearly you can see that there are places where there's high density, so meaning lots of specimens with lots of similarity between species. And 2 locations that are really interesting, of course, would be this area where we have high density, and this one is another representative area. But we can also then, because I can project an individual image into the space, I could kind of carve up that morphospace and just make it represented by the images themselves. So we could look at how those like seemingly kind of arbitrary vectors that we've created, these 2 components, are actually representative of the shapes themselves of the plants. So here I'm just trying to blow that up and give us the most coarse-grained understanding of how that 2048 and 8,000 species, data set and AI model can represent, the diversity of shapes that are contained in our data set. And so the 2 things I did was just throw on some axes onto this plot And then, like by the eye, just looking at how those axes are representative of something biological. So to me, the x-axis really is about the number, the complexity of the divisions of the leaves or the fronds for the ferns. And the y-axis essentially looks like size. So, we have the large divided things off in this quadrant, and large, simpler things over here, and small grass-like things over here, and highly divided small things over here. So it's a kind of a reflecting something biologically interesting about the fact that we wrote all of this data in this model. Yeah, so zooming out then and looking at how that density plot is then overlaid onto this, we can see different areas of the global diversity of our data set where we have high density of species, and then therefore like a representative of common morphotypes. So in this case, this region, which we see densely populated here, contained in this red square, is really things that are very small and not very divided, and we have this other region here that I pointed out. Those are two representative, very species genera of ferns, and so you have the Elaphoglossum, which have this simple representation, and the tree ferns, the Cyathiaceae, which here are represented, a large number of them plotted in the same region and representative of those shapes. So how does shape disparity then track the latitudinal changes in species numbers? We just generated all of this information about the shapes of organisms based on this whole process of developing a classification model, and then basically interrogating it about what the shapes that it had learned that were useful for differentiating species. Now we can just look at how those data are overlaid on the geographic space. So in this case, we looked at how median neighbor distance in the two-dimensional trait space was changing across latitude. And the interesting thing here is showing that species in tropics are certainly more densely packed than outside the tropics. And these dashed lines here are representative of just the tropical boundaries. But also that the significant changes in comparing these pairwise, say moving from the equator north or south, are really happening right across the tropical temperate boundary. So there are what I would say 3 different regimes that are contained, represented by our data set. We have this tropical regime of densely packed species. We have lots of changes happening across the tropical temperate boundary, and then areas that are temperate that are representative of somewhat interchangeably. They're not significantly different from one another once you get outside that area of turnover. And of course, that then just overlaid again on top of that morphological or the total numbers of species. Clearly, you can see that there are major changes happening. Those 3 regimes are potentially associated with the changes that are happening in the globe. So there is some suspicion that we had in our interactions with, especially one of my colleagues, the pteridologist at the Natural History Museum, Eric Schuppeltz. He was really focused on whether or not growth habit played a role, and epiphytism is in particular because of how common it is among these Elaphoglossum ferns. So we have these very simple ferns. They all look very similar to one another, but they have this axis of variation, which is where in the canopy they're able to occupy based on their epiphytic growth. So something interesting there, certainly. Quickly, I just want to talk about this second aspect of what we were looking at, which was how morphological diversity varies according to latitude. In this case, we're looking at kind of the density of trait space. And here, we just wanted to quantify the total amount of trait space occupied along the different latitudes and ask how that varied as well. Here's a method that I actually deployed quite a while ago when I was a graduate student, looking at the diversity of morphospace diversity across different elevations in the Himalayas. This is a method called hypervolume estimation. essentially what we're doing is we're asking how, for a subset of our data set, how our morphospace is occupied, and we can generate then an area, basically a volume that is occupied within our kind of larger morphospace. So here is a representation of that for a band of 0 to 5 degrees north, and then so forth, moving up in latitude toward the poles. So again, we can take our total data set and look at how the volume changes across those different latitudinal bands. You can see that the tropical volumes are really high. But if we really reduce this data set down to like 2 dimensions, there was something very interesting here, which is that there was a little peak in diversity of morphospace, or kind of morphospace occupancy outside the tropics, kind of an extratropical peak in the northern hemisphere. And there are, I guess, lots of things to ascertain from that. I think there's something perhaps going on in Japan and the contributions that Japanese mountains make to some of that diversity. So still more to explore here. And obviously something that I think could yield a lot more interest from the teratology community. Okay, so this project that I'm describing to you is been the product of many years and developing this whole method and approach that we had to a very specific type of data, and that's images that are produced from digital collections. So I was showing you basically one kind of channel through this figure that we put together, where we're collecting some biological material, mounting it, turning that into a set of RGB pixels, and then using what's called a convolutional neural network to process it and generate biological data. But now, of course, there's bioacoustic data and all sorts of other things where we can get to the same types of models, convolutional neural networks. But as you probably know, the natural language models and large language models are now dominating the field in terms of conversations about artificial intelligence. And there's all sorts of exciting and new interesting ways to leverage large language models to kind of achieve similar biological insights. And I just wanted to call out briefly postdoc that I've been working a lot with and who's here working kind of as a research associate at the Field Museum, Mark Ali. Mark has been looking at using text data and generating similar high resolution understanding of the biological diversity contained in textual documents. So we're talking about floristic treatments and monographs and other things that describe the biological diversity of a group. And then here's just an example of something that he's been toying around with, creating dichotomous keys, for example, from what is essentially an unstructured digitized book. And so those are some of the things that we've been processing and thinking about as far as data science at the Smithsonian. And with that, I'll just say many thanks to you all, and I can take any questions and any conversation you want.
Yeah, sorry, I forgot to mention at the outset that Alex has an important Zoom call to take at 1 o'clock, so we did have to break a little early. So we do have, so we want to get him upstairs in time for that, but we do have time for questions. Yeah.
So great talk, Alex. So I have a question about, so when you do so you train on taxonomy, then you get the vector of that you condense into 8 variables, right? So so you're looking at mostly 2 of the variables. I imagine they are driving this divergence, but was any of those 8 variables complete nonsense, something that you couldn't really interpret, or that seemed like the model was picking up something weird about the samples to make taxonomic calls, or did all the make perfect?
Yeah, and that's one of the biggest challenges, I would say. So what you're getting at is we're doing as best we can to disentangle, what most people call the black box of this method. And there is no doubt information that, might be used by, that's embedded somewhere, some bias, right, in our model. That was one of the things that we wanted to, that was why we were pre-processing those images so much, was to try to remove as much as possible anything that wasn't reflective of the shape of an organism. But of course, there's all sorts of other things going on. The color might play a role, and we didn't really want color to play a role in what we were analyzing. I wasn't able to, I think there's a lot more that one can do to interrogate the relationship between things that are present in the images and traits of those organisms that are known from other information that might be present in those 8 dimensions. But no, I don't have any specific, and in fact, I think there are methods now that are, currently being published that are really interesting versions of that low dimensional projection method that take into account a greater interest in reflecting something more like principal components analysis, where you do have a factor loading of what those different components mean and how they're related to the kind of linear measurements of your images. So I think there's a lot to do there, and I think that's the next step with this method for sure.
Thanks, I hope you may hear about this.
Yeah.
Thanks, Alex. The question I had was, so as an implication of this, that firms have no convergence?
They have no convergence.
Or very little, at least at higher levels?
Oh, because we were seeing that that doesn't have to be true. So just because a species is, it isn't true that the genus, so because you're saying the genus level was so accurate. Yeah. Well, that's a good point. Because of course, if you had seen kind of big differences between genus and species, then maybe you would think there was confusion happening at a level of. But I was showing you that most of the conversions or most of the confusion was happening within families, but there were certainly areas where I think that method is actually the place that you would use to look at convergence, because you would say, okay, most of the time we do find that there are, individual taxa kind of occupying unique places in morphospace. But sometimes there are weird things that do get confused a lot, and they're distantly related things. And there are things like that in that visualization. It's just that there are so few that it's more likely, and that's how a taxonomist would operate, right? We would be more likely to confuse something that's closely related, but sometimes there's something that converges, and at this level, it's so coarse, right? That's the other piece of it. It's like you're getting convergence, or you would be studying convergence at a very coarse scale, but there's really interesting ways in which they're differentiating from one another at a higher resolution. Yeah, that's a very good point. I think that's the exciting way to take advantage of that approach, is to look at questions like that.
Hey, I know there's a lot of buzz around AI right now and using it for solving all sorts of problems. Is there places where you think AI does not make sense? Because obviously it doesn't represent knowledge very well. It just, parrots things back at us. So are there places where you wouldn't use either convolutional neural networks or these large language models yet?
Yes, I think that's a very good point. When we started with this project, the idea of using a convolutional neural network was mostly there is an architecture, a statistical framework out there that can take in lots of data and produce for you, if you have the right training data, something interesting, but probabilistic on the other end. And now people are much more used to interacting with models that have been trained on some data set that they don't know anything about, and trying to use that model to then give them back information that they'd like to see. So I think there's two big differences, or that is a very big difference between this approach and leveraging large language models, for example. And I think that any method or approach needs to, particularly define what those training data are and how they bias the results that you get out in the end. That is like the fundamental piece of using AI. And so when the training data are inappropriate for your question, there's just no way that you can get a reasonable or kind of interpretable result. And I think that's the, abundance of caution should be taken, for anybody trying to deploy these methods that isn't thinking very particularly about the data that's going in, how they are biasing anything that you get on the other end. Because even something like this, you might be, I don't know, like somebody kind of naively might be grabbing a model like this and trying to do something interesting with flowering plants, but maybe there's really no good reason to do that if you're not kind of pre-training your model using flowering plants. And so, that's just a naive mistake that someone might make, but those things exist in this kind of methodological framework kind of left and right. And I think there are lots of pitfalls and lots of reason to be cautious about applying AI.
I had a question about following up on John's question about the Fern work. So it seems like there's a lot of interesting kind of downstream analyses you can do with this data. I mean, John kind of hinted at this, like, taking a phylogenetic lens, a geographic lens, like, for example, looking at these morphological hypervolume size and stuff like that. I mean, how does that relate to, like co-occurrence, these hotspots like New Zealand and, in tropics. I'm just kind of curious what other, you guys have delivered stuff in the pipeline.
So, check this piece. So here's just a like one approach to looking at that, behavior habit data that I was looking at. So here's just plotting the differences between something like epiphytic and terrestrial ferns. And so we were looking in the paper really a lot about epiphytism and asking questions about, is that growth habit playing a huge role, and then just comparing hypervolumes between those two. And, epiphytism really is represented by this, what seems to be kind of a small, a very speciose, but very kind of small morphospace. So yes, there are, so many more questions. I think like just the actual approach was, okay, these are, as some of the motivating questions the kind of research I've done in the past. This is a really useful lens for kind of taking on this question of like, how can we produce useful data from this data set that attacks kind of a singular question in biology, but I mean, just in the exact same way that that data can be used for, that these herbarium specimens can be used for all of those questions that people have been studying with them for, decades. It's the same thing, right? So now we have this feature data set, and here we can attack, so many different things, taxonomies, et cetera.
Thanks for the presentation. Just had a very quick point. I think that was related to what was just said here about limitations of artificial intelligence. I thought that was really interesting, a really good question, like you said. So I think the problem might arise when one stops, thinking like a biologist or a scientist, fundamental scientist, start thinking of the problem of AI as an engineer, really. I mean, that's going to make sense in a second. So, for example, when you look at a data set, images, for example, biological data, there is a limit of the amount of information that you can extract from an image. So, for example, you can have images that aren't really holding any information that is biologically meaningful, but to the model, it might, find a way to distinguish between different species, but really you as a human would say, well, this doesn't really make sense biologically. It doesn't know the biology, but it's good at, just like the best analogy is like when you try to understand a word, memorize a word and the meaning of a word. without really looking at the root of the word, I don't know, the Greek or Latin root of the word, but rather you make a stupid analogy in your head. Well, it works, but I mean, how well or informed is it linguistically? Not really. The same goes with models like this. So the question is really always should be, is it aware of the biology, or is it extracting enough biological information, meaningful biological information? Yeah, so that was just the point I was trying to make.
Yeah, it's a good point. I mean, I think we were focused on that from the very beginning, you know, like how to go from black box, it can do the identification, to meaningful biological information or something biological that results from creating a model like this. And, you know, those methods and the approaches that those are in their very infancy in terms of developing those statistical methods for us to know how to leverage those tools. And I think, you know, that's the future of how these tools get used in this discipline, is like developing the statistical approaches to actually generate knowledge about how it reflects biology.
All right, so in order to give Alex time to get upstairs and ready for his next meeting, let's thank him for the talk.
Thank you, Lynn.